Hello everyone, myself Sahana. I am going to present the seminar on test for significance of attributes, test of difference between proportion of success illustration. Let's go with the meaning. A test of significance is a formal procedure for comparing observed data with a claim, also called hypothesis, the truth of which is being assessed. The result of a significant test are expressed in terms of a probability that measures how well the data and the claim agree. Tests of significance of attributes are discussed under following heads. First one, test for number of success. Second one, test for proportion of success. And third one, test for difference between proportions. The topic assigned to me is test for difference between proportion. If two samples were drawn from different population, we may be interested in finding out whether the difference between the proportion of success is significant or not. In such a case, we take the hypothesis that the difference between the proportion of success in another sample is due fluctuation of random sampling. The standard error of the difference between proportion is calculated by applying the following formula. Z is equal to P1 minus P2 divided by square root of P1 into Q1 divided by N1 plus P2 into Q2 divided by N2. Here P1 is the proportion of success in sample 1 and P2 is the proportion of success in sample 2, and Q1 is the proportion of failure in sample 1 and Q2 is the proportion of failure in sample 2. N1 is the size of sample 1 and N2 is the size of sample 2. If two samples are drawn from different population, then we might know the difference of the proportion of success is significant or not. In such a type of situation, the null hypothesis will be HA subset P1 is equal to P2. It implies that two proportions are same. HA P1 is not equal to P2. It implies two proportions are different. Either P1 is less than P2 or P1 is greater than P2. Here P1 is the proportion of success in sample 1 and P2 is the proportion of success in sample 2. Now let's see the statistic in the case test of can be given by the following equation. Z is equal to P1 minus P2 divided by square root of P1 into Q1 divided by N1 plus P2 into Q2 divided by N2. Here P1 is the proportion of success in sample 1 and P2 is the proportion of success in sample 2. Now let's see the practical problem to understand the application of hypothesis testing in case of testing the difference of proportion. Problem: a drug research experiment unit is testing two drugs newly developed to reduce blood pressure level. The drugs are administered to no different sets of animals. It means tested on different groups of animals. In group 1, 350 of 600 animals tested in on drug 1, and in group 2, 360 of 500 animals tested its own drug 2. The research unit wants to test whether this is a difference between the efficacy of the set two drugs at 5% significance level. Efficacy implies the effectiveness of drug for medical treatment. Hence, for the problem, can we conclude that the effectiveness of the two drugs is different at 5% significant level. Now, to deal with this problem, first state null hypothesis would be H1 subset P1 is equal to P2. It implies the efficacy of two drugs is same. alternative hypothesis HA P1 is not equal to P2, it implies the effectiveness of two drugs are different. In this problem, N1 is equal to 600, N2 is equal to 500. It refers to size of the sample. Proportion of success in sample is calculated as P1 350 by 600 is equal to 0.583 and proportion of failure Q1 is equal to 1 minus P1 is equal to 1 minus 0.583. We get 0.417. Proportion of success in sample 2 is P2 is equal to 360 by 500. We get 0.520 and Q2 is equal to 1 minus P2. We get 1 minus 0.52. This will come out as 0.480. Now, replacing the value P1 as 0.583, Q1 as 0.417 and P2 as 0.520, Q2 as 0.480 and N1 as 600 and N2 as 500. Let's apply the formula. Z is equal to P1 minus P2 divided by square root of P1 into Q1 divided by N1 plus P2 into Q2 divided by N2. We know P1 as 0.583 minus P2 0.520 divided by square root of P1 0.583 into 0.417 divided by 600 plus 0.520 into 0.480 divided by 500. After subtracting 0.520 from 0.583, we get 0.063 divided by square root of 0.243 divided by 600 plus 0.250 divided by 500. We get 0.063 divided by 0.03 is equal to 2.1. And after processing all the calculation, the value of Z come out as 2.1. Conclusion for the problem. Here HA subset P1 is not equal to P2. Rejection region applying two-tailed test at 5% significant level. The rejection region of the table under normal curve Z distribution find that Z is greater than 1.96. Now our calculated value Z is 2.1, which is greater than the critical value of Z. Hence, the null hypothesis will be rejection region. Therefore, the null hypothesis is rejected, and we can conclude that the efficacy or effectiveness of two drugs are different significantly. Conclusion for the topic. A two-sample Z test is used to test difference between the two population proportions P1 and P2 when a sample is randomly selected from each populations. Thank you.